inheritance. Now today we will see how this Mendelian and non-Mendelian inheritance holds good to the chromosomes or to the cell. So today we will discuss another new chapter, that is chromosomal theory of inheritance. Now how this inheritance is based upon the behavior and the structure of chromosomes. So the behavior of chromosome is very crucial for the inheritance of characters. So today we will discuss the chromosomal theory of inheritance. So let us go. Now the chromosomal theory of inheritance says that this is the chromosome on which the different genes or the different characters are present, and they are inherited from one generation to another generation as the chromosome goes from one generation to another generation. So we will check what are the salient features and what are the important points of chromosomal theory of inheritance. Now it says that the chromosomal theory of inheritance was proposed by W.S. Sutton and T. Boveri in 1902, and it was confirmed by T.H. Morgan in 1933. So many scientists, they have experimented on various organisms, and finally they have put forth this chromosomal theory of inheritance. This looks very complicated, or it sounds very complicated, but this is very simple. What it says, that the genes are arranged linearly on the chromosome, and the genes are inherited from one generation to another generation along with the chromosomes. So as proposed by Sutton and Boveri and confirmed by Morgan, what it says, that chromosomes are the carriers of hereditary information. It possesses the Mendelian factors, or in other words the genes, that segregate and assort independently during transmission from one generation to another generation. So what we have discussed in Mendelian inheritance, that the famous two laws, one is the law of segregation, another one is the law of independent assortment. So these two laws, one is segregation, the another one is independent assortment. It holds good only because of the behavior of chromosomes. Now in a diploid cell or in a diploid organism, now there are two alike chromosomes which are called as homologous chromosomes or homologous pair. One chromosome comes from the maternal side, the other one comes from the paternal side. Now these two chromosomes has similar types of genes and similar numbers of genes on them, and they segregate from each other during the time of gamete formation. then the independent assertly, that means each gene and the alternative forms, they have an independent chance to assert or to make a combination with the other forms of the another gene. So, the chromosomes are the carriers of the hereditary information and they possess the Mendelian factors or genes over them, and they show the segregation independent assortment during transmission from one generation to another generation. Now, so chromosomes are the physical basis of heredity. Now, we cannot see a gene how they are inherited, but the physical basis that we can see is only the chromosome, that we can see even if by using a compound microscope. Okay. So, we can see the chromosome, how they are segregating, how they are forming, or how a cell is being, it is forming two daughter cells. Okay. So, the behavior of chromosome, how they are being arranged on the equatorial plate, how they are being segregated to two different poles or to two opposite poles, and they form two clusters to form two daughter cells. So, this is the physical basis of heredity. So, they have a special organization, individuality, functions, and capable of self duplication. Now, a chromosome can self duplicate itself. We will discuss it later on different or on upcoming slides, that how a chromosome after division it becomes a single chromatid, and that single chromatid it replicates or it duplicates during the S phase of the cell cycle to make it a full fledged chromosome having two chromatids, and these two chromatids will segregate from each other at the time of the division of the cell. Okay. So, chromosomes play a vital role in heredity, mutation, variation, and evolutionary development. So, this is very important that the chromosome play a vital role in heredity, mutation, variation, and evolutionary development. So, if the chromosomes would not be there, then there would be no heredity, or there would be no inheritance of character from one generation to another generation. So, if any changes occur because of some reason or the other, and most importantly because of mutation, so then the gene or the different Mendelian factors, they vary from each other, and this variation is crucial, or it is the key to the evolutionary development. And this is because of the changes, because of the mutation, because of the variation, because of the natural selection, this evolution of different species has taken place till now. Okay. So, the chromosomal theory of inheritance is that important. So, let us have a look at the salient features of the chromosomal theory of inheritance. So, the salient points in support of the theory says there is a link between one generation and the next is through male and female gametes. The male and female gametes have half of the number of chromosomes, isn't it? So, the sperm or the egg and the two must carry all genetic or hereditary traits or characters. Both the gametes contribute equally in the heredity of the offspring, and nucleus possesses now in which the chromosome is present. Now, the chromosome is present inside the nucleus, okay, as chromatin reticulum or whatever it may be, but the chromosomes are always confined within the nucleus in an eukaryotic organism. Hence, chromosome must carry the hereditary traits, because we know that at the time of cell division, the nucleus also divides into two nuclei, and these two nuclei acquire some cytoplasm and they becomes the two daughter cells. So, loss of a complete or part of a chromosome produces structural and functional deficiency in the organism. So, these are somewhat like the chromosomal aberration. Let us suppose there are two chromosomes. For example, if I will draw a picture here, Let us suppose two homologous chromosomes are there like this. Okay. Now, for some reason or the other, this portion is discarded. Let us suppose this portion is discarded. Now, what will happen? Now, if this portion is deleted, this is the portion, okay, which is to be deleted. Now, what will happen? The genes that would be present over here, okay, now they will not be there, okay. So, that means some portion is deleted, means it is creating some kind of aberration or some kind of functional disparity. So, that has been written here, that there might be a structural and also functional deficiency. Let us suppose by some means or the other, the total chromosome is being deleted. Well, so obviously this will also, because this will also cause a partial deficiency in that gamete or in that organism. So, always it brings any kind of changes, either partial loss or complete loss, it brings structural and functional disparity or deficiency in the organism for valid reason. Now, like the hereditary traits, the chromosomes retain their numbers, structure and individuality throughout the life of an organism and from generation to generation. The two neither get lost or mixed up. Chromosomes never get lost or mixed up, okay, and they behave as units. Both chromosomes as well as genes occur in pairs in the somatic or diploid cells. Let us have a look what it says. So, now you see how it happens. Now, where lies the chromosome? Now, if this is a cell, so inside the nucleus you have the chromosomes. They are in the form of a reticular structure. So, you cannot view it directly. When you can view, it is only at the stage of metaphase when you can see the complete typical structure of a chromosome. So, what you will see there, that you may find two pairs of chromosome at the time of meiosis, or a single pair of chromosome at the time of meiosis, and this is the homologous chromosome. Okay, and any loss that occurs to this is a partial or structural and functional dysfunctionality that happens, or deficiency happens to the organism. And as you know, it always occurs in pairs, and during the time of gamete formation it will have a single chromosome, okay, either this or that, not both. So, a single gamete, either it will be a male gamete or a female gamete, it will have only a single chromosome out of these two homologous chromosomes. So, both chromosomes as well as genes, they occur in pairs. Why they occur in pairs? Because we have two homologous chromosomes, as already I have told you that if I will choose any locus, okay, so on this locus I will get the Mendelian factor, or in other words, what is that? Gene. Only at this locus, this gene, a single gene will be found at this locus, and obviously it will occur in pairs. For example, like capital T or small t or capital T, small t, or small t capital T. So, these 4 combinations I will get, but what it will be? Now, this will be 2 numbers of genes, okay, or at this locus I will find it maximally 2. So, at the time of gamete formation I will only get a single one, a single gene or the single form of a gene, okay. So, next slide. Now, this says the same thing, a gamete contains only one chromosome, that I just said it right now. It contains only one chromosome of a type and only one of the 2 alleles, that I just told you, that either it will have capital T or small t when it comes to gamete, but when it comes to organism they may have both of the genes or both of the alternative forms of the same gene. Now, the paired condition of both chromosomes as well as Mendelian factors is restored during fertilization. So, at the time of gamete formation there will be a single gene form, okay. Now, when fertilization occurs, what happens? Now, a male gamete fuses with the female gamete. So, here a chromosome from the male gamete and a single chromosome from the female gamete, they both of them together they form a pair, and this becomes a homologous pair of chromosome. So, that is why during fertilization this pairing occurs and we regain the same numbers of chromosomes as it is in the diploid organism. So, the paired condition of both chromosome as well as genes is restored when? During fertilization. And the genetic homogeneity or heterogeneity, that is, or the dominance and recessiveness can be predicted by chromosomal type and behavior. For example, whether it will be homozygous like capital T, small t, or it will be heterozygous like capital T, small t. This will be heterozygous, this too will be homozygous. So, these are all predicted by the chromosome type and their behavior. Sorry. Then homologous chromosome undergo synapses during meiosis. This all of you know it very well, that this exchange of chromatids happens only at the time of crossing over. So, crossing over takes place unless and until there will be pairing of chromosomes, okay, and that pairing is called as synapses. So, when synapses occurs, when one chromosome or the 2 homologous chromosome, one from the maternal side, another from the paternal side, they come closer to each other and they form a pairing. So, this is called as synapses, and this synapses decides that what kind of chromosomal exchange or the segment exchange will occur. So, homologous chromosome undergo synapses during meiosis, not during mitosis. So, and then separate or segregate independently into different cells, which establishes the quantitative basis for segregation and independent assertment of hereditary factors. So, that will decide whether these 2 gametes they will, or the 2 gene forms, they will segregate from each other, or after that the different forms they will assort independently with each other. So, these are all decided during the process of meiosis, and synapses of course it helps in this prediction. So, in many organisms sex of an individual is determined by specific chromosomes, and this is called as sex chromosome. Okay. So, the sex chromosome decides, so the sex chromosome decides the sex of an organism. For example, if we have a X chromosome or 2 XX chromosome, it decides that this is a female organism, whereas the X and Y, this decide that this is a male organism. So, this is female. So, the femaleness or this your sex is determined by this X chromosome, and the Y chromosome it decides the organism to be male. Okay? And finally, the biochemical studies reveal that genes or the hereditary units are composed of DNA in eukaryotes and RNA in some prokaryotes. Okay? And the major amount of the DNA is found in chromosomes proves beyond any doubt that chromosomes are the carriers of hereditary units that we call the genes. Okay? So, before going to that, this is very simple that so this says that this is very simple in very simple terms. Now, a single chromosome, let us suppose we will draw a linear view. What the Morgan has said that if this is the two pair of homologous chromosome, okay, now the Mendelian factors or the genes, whatever you may call it as, they are arranged how? They are arranged linearly on different locus, and the locus position is fixed for both of the chromosome. So, you see the genes are arranged linearly over the chromosomes, and you can see that I am drawing both of the locus or loci on the same point on the chromosomes. So, the biochemical study says that this is the different numbers of genes, as I have pointed many slanting or slipping lines. Now, all the slipping lines shows that these are the positions of different genes and they are arranged linearly over the chromosome. So, at the time of gamete formation, so these two chromosomes, they segregate from each other. So as the genes are arranged over the chromosome, so the genes also segregate from each other for a particular locus, but they may assort independently with the different forms of other genes like this. Let us suppose this is one gene, now it can assert independently with this two, with this two, with this two, with this two. So let us go for a diagrammatic expression. So this is one diagrammatic expression. For example, yellow seed and green seed, round seed and wrinkle seed. Now you see, this is a homologous chromosome where you have this capital Y and small y, then round seed and wrinkle seed, capital R and small r. Okay. Now the chromosome replicates, isn't it? Now this is a single chromatid structure. Now when it replicates, what happens? Now this replicates to this. You see? Now this is capital Y, this will replicate, the small y will replicate to this. This will be small y and small y. So similarly, the capital R, this one will replicate like this and this will replicate like this. Okay? Now as the replication occurs, now the orientation can be in two different ways. Now you see, this is one capital Y and capital R are present on the same orientation. But here you see capital Y and small r are presented at one orientation, whereas the other two on the other orientation. So what will happen? So during the time of meiosis, now what will happen? This portion will come as a single unit, okay, and this portion will come as a single unit like this. Well, similarly this will come as a single unit and this will come as a single unit, okay. Now what will happen? So we will have here what? Capital Y, capital R and capital R. Here what we have? Capital Y, small r and small r. Well, this one will be capital Y, sorry, this one will be small y, okay. This one will be capital Y with small r and small r. This one will be small y with capital R and capital R. So this is regarding meiosis 1. Now when it becomes meiosis 2, now what happens? Now this one will come like this, okay. So this two chromatids, they will separate from each other as it happens in mitosis. So as it is meiosis 2, so this will split up like this longitudinally. So one capital Y will come to this place and the capital R will come to this place. So as the different things. So here what you are going to get? 1/4 capital YR, 1/4 small y, 1/4 capital Y, smaller, and 1/4 small y, smaller. So whatever salient features we have studied or we have discussed or we have read, that is depicted in this diagram, okay, showing that at the time of gamete formation, the genes or the Mendelian factors, they segregate from each other, so as the chromosomes, and then they independent assort or they independently assort with each other, okay, so as the chromosomes. So this is the thing that we have discussed. So this is the clear cut picture of showing the chromosomal behavior and how the gene present on the chromosome show the similar kind of behavior, okay. So next slide we will see or we will discuss the structure of chromosome. What are the different structure of chromosome and how they are being, where they are situated and who has discovered this and all. So first of all, you see chromosomes are thread like structures located inside the nucleus of an eukaryotic cell. So if I will tell you now, if I will draw a diagram, so if it is, this is nucleus, the chromosomes, as these are thread, they are very much intermingled and you cannot see the individual thread as chromosome. So when you will see this during the phase of prophase, where inside the nucleus from the very beginning of prophase, you will see that this thread are, these interwoven threads are slowly they are separating from each other. So you can see this thread like structure, and finally with this disintegration of nucleus, you will only see the chromosomes during what? During prophase as thread like structure, but during metaphase you will see this kind of structure like two chromatid structure, okay. So each chromosome is made up of protein and a single molecule of DNA passed from parents to offsprings. And how the DNA is present? The DNA is a double stranded helical structure and there is a single molecule in a single chromatid. So if there would be two chromatids in a chromosome, then it will have two molecules of double stranded DNA, okay. The term chromosome comes from the Greek word chroma, means color, and body means soma. Now the chromosome takes staining, particularly this many kind of staining it takes. So as this takes staining, it becomes colored. Okay, so that's why they are called as colored body. So the historical information says the term chromosome was discovered by Walter Fleming in 1882, and the term chromosome was coined by Waldier in 1888. Now regarding the structure of a chromosome, the structure is very simple. If I draw a single chromosome or a full-fledged chromosome having 2 chromatids, now you see, now this is the centromeric region, okay, these are the chromatids, these 2 are the chromatids. We will see the picture. This is the centromere, and these end regions, these regions, okay. So one at this point and the other at this point. So these 4 regions are called as what? Telomeres. So these are the most important points. Let's see. Each eukaryotic chromosome consists of a long linear DNA molecule associated with proteins. Compact complex of proteins and DNA are called chromatin. We will discuss it, what is the structure of chromatin. So the DNA molecule, when it wraps over the histone protein, so what kind of proteins we have, we will discuss it. So when the DNA molecules are wrapped over this histone proteins and it form a nucleosome, then the nucleosome further being packed and coiled to give you a chromatin structure, and these chromatins are highly packed within this chromosome or within the structure which is known as chromosome. Chromatin contains the vast majority of DNA of an organism, okay, and eukaryotic cell possess multiple, that means many numbers, large linear chromosomes contained in the cell's nucleus, okay. So the eukaryotic cells, whereas in contrast, what does the prokaryotic cell have? Now the prokaryotic cell, they have a single circular naked DNA. This is not like the DNA is wrapped over the histone proteins and all, but they have a single circular double stranded DNA, whereas in case of eukaryotes, we have many numbers of linear chromosomes which are present in 2 sets, okay. So the structure of a typical metaphase chromosome has, as I have already have told you, a centromere, a telomere region, then the chromatids, then there is a short arm and there is a long arm. So let us have a look at this. Now you can see from this portion to this portion, this 3, this is the short arm, okay. Then from this to this portion, the 4 is the long arm. This middle point, obviously the centromere, and these ends, okay, these ends are the telomeres. This is also the same, these are the telomeres, and this is the chromatid, this is the chromatid and this is the chromatid, okay. So these 2 are the chromatids, the 2 chromatids, okay. Now you can see a single long strand of DNA, how it has been packed into such structure, okay. Now what is centromere? Centromere is the specialized DNA sequence, okay, of a chromosome that links a pair of sister chromatids. As I told you in this one, So, as you can see, now this is one chromatid, okay, this is another chromatid. Well, and these two are what? These two are sister chromatids. These two are sister chromatids. These two are sister chromatids. So, what it says that the centromere is a specialized DNA sequence of a chromosome that holds or that connects two sister chromatids. Now, during mitosis, the spindle fibers attaches to which portion? To the centromere via the kinetochore. Centromeres were first thought to have some genetic loci that direct the behavior of chromosomes, but mostly the centromere does not have such kind of DNA sequence which have some genetic loci or some Mendelian factors or the genes. Generally, it is devoid of genes and is packed with your heterochromatin like structure. So, next is the concept of chromatin. So, what is a chromatin? So, chromatin, as we have discussed, now what are the different structure? Now, one is centromere, telomere, the two chromatids. So, the chromatids, they have the chromatin structure or that chromatin thread. So, chromatin is a complex of DNA and proteins that forms chromosomes within the nucleus of an eukaryotic cell. So, chromatin is a complex of DNA and that thread like structure is known as the chromatin. Now, regarding the function, the primary function of the chromatin is to package the long DNA molecule into a much compact denser form. The denser form means now generally at the time of prophase, we can see that the chromosome comes as a thread like structure. So, if this is the centromere, this is the chromatin threads like, and the chromosome looks like this, but because of this packaging of the chromatins, now we will see the most compact phase of the chromosome at the metaphase stage, where it is highly compact and it prevents any type of tangled or any type of entanglement with each other. As we have shown you here that this is the compactness that is being shown by the chromatin structure. So, this prevents the strands from becoming tangled. It plays important roles in reinforcing the DNA during cell division and it prevents DNA damage. So, important is the function of chromatin. Now, regulating, it also regulates gene expression and DNA replication, and it facilitates proper segregation of chromosomes during cell division. Once again, regarding the function, the primary function is to pack the DNA into a highly compact and dense structure, and it prevents, this kind of packing prevents the long DNA molecule to get entangled with each other. Well, now next it plays an important role in reinforcing the DNA during cell division. Then it prevents DNA damage, it regulates the DNA replication and gene expression. Well, and finally it facilitates the proper segregation of the chromosomes during cell division. So, this is about the structure and function of chromatin. We will see how the long strand of DNA, which may extend into kilometer long, how they are being packed into a very tiny nanometer structure like the chromosomes. So, this is a clear cut picture. Now, you can see the DNA molecule, which is a long double stranded molecule. So, how it will be packed for the first time. Now, this DNA, they will be wrapped over the histone proteins. Well, So if this is a single molecule of DNA, so it will be wrapped over the histone proteins like this, well, and it will form the chromatin structure like this. So it will form a nucleosome. So I will explain you how this is being formed. First of all, let us see the diagram. So these are the histone proteins. The histone proteins forms a core. Now how many histone proteins are involved? Now there are 4 histones are involved. One is H2A, H2B, H3 and H4. Now they forms an octameric structure. So 2 tiers of tetrameric structure, they are being superimposed upon each other, over which the DNA get wrapped, taking a 1.4 ton. Well, and this is being wrapped like this. Now the DNA molecule comes, it gets wrapped, then again like this, then like this, okay. So like this. So it gets wrapped over like this. Now the DNA wraps around histones, forming what? Nucleosomes. Then the nucleosomes condenses into a chromatin fiber. So the nucleosome then condenses into a chromatin fiber. So what happens here? This nucleosome, it get condensed. Well, so when it condenses, now it becomes more and more packed and compact. Okay. So what happens? Now this is the, so this will becomes much compressed. Well, so forming the chromatin fiber. So the chromatin fiber further condenses. Ahuri say adhika adhika condensed haba. Okay. So it further condenses to give you a structure. Now this condensed chromatin forms a chromosome. So now you see, first of all there is a simple double stranded long large DNA molecule which gets wrapped over the histone proteins. Well, then this histone proteins, it form a nucleosome. So this nucleosome, they get themselves compressed. Okay, this compression creates the chromatin fiber. The chromatin fiber further condenses to form this kind of structure known as chromosomes, having 2 chromatids. Okay, let us have another picture. Now you see here, now this is the DNA molecule, it has wrapped over the core of 8 histone molecules. Well, and there is a linker DNA which connects between the 2 nucleosomes. Well, and this linker DNA is always associated with histone H1, and this nucleosome then further it condenses to form this chromatin structure, then which is of 30 nanometer. Okay. Now you see this DNA is 2 nanometer long, this what the diameter, then this becomes a 30 nanometer long chromatin fiber. It further condenses to give you a 300 nanometer long much packed chromatin, and finally it forms a 700 nanometer long chromatin packed structure. Finally it becomes a 1400 nanometer chromosome structure. So before going to anything, let me tell you
ऑफ ହେଇଥିଲା ବରା
पूरा ऑफ ହେଇଥିଲା
डिजाइन फ्लैट शो
So you see, now this is the packaging of DNA. Now the DNA, it wraps over histone proteins. Now let me tell you how the histone proteins are arranged. Now the histone forms an octamer. So the histone forms an octamer structure with the help of 4 histones. One is H2A, H2B, H3 and H4, and 2 such tires are being formed, one at the front, the other one at back, and the DNA molecule, it takes a 1.3 ton, then again now it wraps over another histone octamer. Okay, like this. Now in the due course, this is the linker DNA. This linker DNA is associated with H1 histone protein. Okay, then it again wraps over the same octamer. Then again another octamer is there like this. So it take another turn like this. So and it is always associated with H1 protein. So these are all histone proteins. Again here it is another H1 proteins. So this is being explained here that how a DNA is wrapped over the histone proteins like this, and this structures, these all structures, they are called as what? Nucleosomes. Okay, so these nucleosomes, they condenses to form the chromatin structure or the chromatin fiber, which further condenses to give you a structure like chromosomes having 2 sister chromatids. So this is another picture how it is get packed. So here you can see the clear cut picture. Now this is the core of a 8 histone molecules. So what are the 8 histone molecules? So the 8 histone molecules are H2A, H2B, H3 and H4, all in 2 numbers. And besides that, the linker DNA, it is associated with what? The linker DNA is associated with H1 histone protein. So this is the figure. So it is there in order to what? Fix the wrapping of the DNA over this octamer or 8 histone molecules, and it forms the nucleosome, which further condenses to give you the chromatin fiber. So it pack into a coil that twists into another larger coil, and it produces the chromatin fibers. The coils fold to form loops. So this folds to form the loops. So these are the loops, okay, and the loops coil over further forming a chromosome structure. So we have, as I told you, that in an eukaryotic cell we have many numbers of linear chromosomes. Now you see, this is a typical metaphasic chromosomes, and mostly they are present on the diploid number. So and regarding the telomere, now the telomere is the end of a chromosome. Telomeres are made up of repetitive sequence of non-coding DNA that protects the chromosomes from damage, and each time a cell divides, the telomeres become shortened, okay, and eventually the telomeres become so short that the cell can no longer divide. So it is only because of the telomere that helps in the process of cell division. So in the next class we will continue further regarding this packaging of DNA into the chromosomal structure. So thanking you all, have a nice day. Cell division. So in the next class we will continue further regarding this packaging of DNA into the chromosomal structure.